Okay, welcome back to the Classics of Immunology Journal Club. Before I get started, I want to remind you to like the video if you liked it, and click on the bell to get notified for more, and check out the website, my website, and go onto the writing tab, and you'll find all the classic papers in immunology there. Also, check out my social media sites because I tweet a lot, and you might pick up on some pearls of wisdom, one would hope. Today, we're continuing on in the saga of how T cells help B cells to make antibody. As you recall, if you go back in the journal club, it was first found out in the late 60s that if you took T cells away from B cells from splenocytes, mouse splenocytes, for example, put antigen in, and everybody used sheep red blood cells, you didn't get any antibody. And that was the beginning of T cell help. So the question is, now here we're 20 years later in the 80s, and we're just beginning to find out how T cells help B cells. And the paper that we're going to review today is a continuation of a paper that last time we had the journal club of Junichi Ohara and Bill Paul, who were working with a T cell derived factor that they called B cell growth factor. They'd made a monoclonal antibody to it. This week we're in 1986. And we're at a paper that came out in Nature in February of that year. The authors, there's multiple authors, but I just want to highlight a few. Yoshihiko Noma, who was a postdoc in Tasuku Honjo's lab at the University of Osaka in Japan. And then Paschalis Sideras and Eva Severinson, who were from the Karolinska Institute. And this was a collaboration between Honjo's lab and Severinson's lab. And the title of this paper is The Cloning of the cDNA Encoding the Murine IgG1 Induction Factor by a Novel Strategy Using the SP6 Promoter. And this basically became Interleukin-4, and that's how we should title this particular journal club entry, Interleukin-4, because it became a thing as a consequence of this paper. So the background on this, as I said, this is a collaboration between Honjo's lab and Eva Severinson's lab. They both added their particular expertise, as we'll get into. Honjo being the molecular biologist trained in Phil Leader's lab at the NIH back when he was a young blade. And by the time this time came around, he was an established professor at Osaka University. And then he subsequently switched to Kyoto University. In the introduction of this paper, they point out that there are 8 different constant regions, genes, in the genome that code for the constant regions of the different immunoglobulin molecules that gave them their definition of the isotype. And of course, the constant region then, although it didn't bind antigen, the constant region was the one thing that determined the biological activity of the antibody in vivo by its ability to interact with primarily macrophages, phagocytic cells through the Fc receptor, and other kinds of things. And so the question was, Honjo had already established the order of these genes in the genome, and he became established as somebody to pay attention to as a consequence of that whole business. The other thing that they had shown was that and what was known at the time was that B cells, if you just looked at naive B cells in the lymph node or the spleen or in the peripheral blood and so forth, they had surface immunoglobulin with the heavy chain of the IgM, the mu chain, and or IgD. But they did not necessarily, most of the cells did not necessarily have IgG on the cell surface, and they certainly didn't have any IgA or IgE. So there was an idea is that there was something going on that somehow the genes played funny games and rearranged their expression, and it wasn't really understood how they did that. And that's why both Severinson and Honjo were interested in this particular phenomenon. And it was known that this could be where some of the T cell help took place. in the generation of B cells. And that is, the isotype would switch from the mu chain constant region to one of the IgGs or IgA or IgE. The other things that had been established was that there were a couple of papers that came out in the Journal of Experimental Medicine in 1982. Now we're in 86 now. Ellen Vitetta from the University of Texas Southwestern at Dallas had shown that there was a T cell derived factor that would cause B cells to switch most of their immunoglobulin production to IgG1. IgG1 in the mouse was the primary IgG that was found in the plasma, and so that was considered an important thing that this T cell derived factor could do. So they called this the G1 switch factor. And at the same time, and in the same issue of the JEM, Bill Paul's group had shown that if you took, actually Vitetta's group used lipopolysaccharide to activate spleen cells and then threw in the factor to get them to switch to gamma 1 rather than IgM. Bill Paul's group used a very similar assay, but a little bit different in that they didn't activate the B cells with LPS. Rather, they activated the B cells with anti-mu, an antibody to the mu constant region. And if they added then their T cell derived factors, whatever they were, they could get the cells to start to proliferate. And so they thought, well, we're looking at a different kind of a factor. We're looking at a B cell growth factor. So the question is that how many factors are there that T cells use to help B cells? And so let's see if we can sort this out. Now, at the time, it was very difficult to get enough protein from any source to be able to purify enough. And I've gone through a lot of that in terms talking about the IL-2 story and so forth. And so that avenue, by 1986, it was clear that the best way to go about trying to figure this whole situation out was to see if you could clone the cDNA that would be encoding for the protein, rather than trying to generate lots of protein and then purify the protein. And so that's what Honjo was good at. And Severinson was really interested in this particular question. And she had a couple of papers the previous year in 1985 in the European Journal of Immunology, where she had established or generated a very quantitative, nice assay for this IgG1 switch factor. And so they put their two expertises together to do these experiments. So in the materials and methods of this paper, which is primarily in the figure captions and the table captions and very tiny little print that you have to use a magnifying glass to figure it out, the quantitative assay that Eva Severinson had worked out was taking B cells from spleen, either purified or unpurified, activating those B cells with LPS, and then titering in the same kind of fashion we did with the IL-2, the TCGF assay, and they could get very nice dilution curves with that sort of thing. And she had worked out previously when she was a student in the early 70s at the Karolinska Institute, a graduate student. And one of the things that she had done was to work out a plaque assay that you could show that was isotype specific for the different immunoglobulin isotypes. And so they used that assay to look for IgG1. And they could find a dose-dependent increase in the number of plaque-forming cells for IgG1. The other thing that Ava did was that she made an IL-2 dependent T cell line. Actually, she got that out of the back of the incubator, she says, subsequently, when she visited Rob McDonald's group down at in Lausanne and at the Ludwig Cancer Institute. And this cell line ultimately was designated with just 2.19 was the name of it. And they found that when they activated this cell line with concanavalin A, they could get lots of this IgG1 switch factor based on her assay. And she then grew up 10 to the 9th cells, which was a tour de force for this kind of a thing, because she needed IL-2 to grow the cells up and so forth. She extracted the RNA and she sent that off to Honjo's lab. And then Honjo purified out the messenger RNA, or maybe they did that in Stockholm, I'm not sure. But Honjo also put the poly A plus, or the messenger RNA, over a sucrose gradient, and took fractions of that, and then tested this subsequently to try to find out whether or not they had the right messenger RNA. What Honjo did then was, and so that was what Severinson's contribution was in the whole thing. What Honjo did then was he used the IL-2 clone, cDNA clone, to work up his system to see if he could use the biological assay system. That's what Severinson had, together with the molecular biology, to see if you could detect the protein that was causing this IgG1 switch. And after several attempts using different kinds of systems, they hit on something that had just been published a year previously from Tom Maniatis' group at Harvard, where they could use a special RNA polymerase called SP6 polymerase, and they could do an in vitro transcription, so that they could transcribe RNA from cDNA in vitro. And then they would need a way to make protein, to translate protein from the RNA. And so they used the frog oocyte assay, which had been set up in his lab and many other molecular biology labs at this point in time. And then they assayed the protein that they got out of the frog oocytes in the biological assay that Severinson had set up. So the results of these experiments, and I'll show you in the screen share. In table 1, this is the IgG1 inducing activities directed by the SP6 cDNA clones. And on here, over on the right-hand side of this table, is the number of plaque-forming cells per culture. They could get plaque-forming cells out of the total cDNA library, and it became positive, whereas if they used the wrong restriction enzyme that cut the protein or the messenger RNA, it would be 0. The positive control were supernatants from the 2.19 cell line. That was positive. Here's the sucrose gradient fractionation of the cDNA library, and the fraction, there was one particular fraction that had most of the activity, fraction 9, and it was positive. Positive control was positive. Then they cloned and recloned and recloned, subcloned these libraries, and they came up with 2 clones that were individual clones that were positive in the bioassay, clone number 293, and then clone number 374. And those were the cDNA clones that they used to take further in their experiments. This figure shows the results of their cloning efforts. In the upper is the restriction enzyme map of the gene, and actually this is the clone number 374. They found that the predicted amino acid sequence, which is shown here, so we've got the predicted amino acids here, and then on the lower column is the cDNA sequence, and you can see that this gene has all the things that you would hope for: a 5' untranslated region, an ATG start site, a 3' untranslated, a poly A tail, and it had a signal sequence of about, I think, 29 amino acids. So this looked like a pretty bona fide cytokine gene, even though there hadn't been too many cloned out at this particular point in time. They went into the libraries and they found that there was some homology with a sequence of the GM-CSF cDNA, couple actually, and then also with a gamma interferon cDNA or protein sequence. And so they figured that they were on the right track on this whole thing. And this then shows this same clone 374, and that was then used to make messenger RNA and then translated in the oocyte system. And you can see that the in figure 3 here, the positive control is in the open circles, and that was soups from the 2.19 cell line. Then the positive clone and the RNA and then the protein translated from the RNA are in the solid circles, and you can see this nice dilution curve. These are doubling dilutions of the soups from the oocytes or from the T cells. The triangles are a negative clone, and the diamond down here are no supernatants whatsoever. So this is the number of plaque-forming cells per culture, the IgG1 plaques, then IgG3 plaques, and you can see that the same protein that caused the cells to make IgG1 inhibited the production of IgG3 and IgG2b, and that was one of the questions that they wanted to find out, was whether or not this is due to the same T cell factor or a different T cell factor. There was no effect on IgM in these experiments. And so they figured they were on the right track. And so the other thing that was known at the time was about this B cell growth factor that Bill Paul's group had talked about. And it's the same symbols here in this figure, so that the solid circles are the positive clone and the open circles are the T cell soups from the T cell line. And you can see this is at I think 10 micrograms, and this is at 2. And so that you can see that the same thing holds true. The assay here is anti-mu activation of the B cells, blasts, and then their proliferative response in terms of counts per minute of tritiated thymidine incorporation. And again, as you dilute, this is increasing amounts of supernatant, you get a nice dilution curve. So then the last thing that they looked at was whether or not this same protein would cause the cells to increase their expression of class 2 MHC gene products. And this is a flow cytometry of anti-class 2 here with 2 different clones. And you can see that was positive. So now the same protein can do 3 different things. It can cause IgG1 switch, it can cause B cell growth, and it can cause an increased expression of class 2 MHC. So I'll stop there. The size, this is forward and side scatter on the flow cytometry, and the cells didn't change during the assay period. So it was just an increased expression of the class 2. So on to the discussion of this paper. This particular paper then solved, several different questions about how many how T cell derived B cell factors are there, and in this situation we got one factor or one gene that would code for a protein that would do 3 different things to these B cells. And on the basis of that, they decided that this was enough criteria that they could call this interleukin-4. So this is really in the early days of the interleukins, or at least the chemical definition and the biological definition of these interleukins, because we only had interleukin-1 and interleukin-2, and then interleukin-3 I haven't talked about. Turns out that that's a hematopoietic cell growth factor. And so it wasn't really a lymphocyte, a lymphokine, an interleukin that helped the lymphocyte category. And so it got sort of short shrift in the immunologic literature, but it got more attention by the hematopoietic people, the hematology types. So that's it. And so this was interleukin-4, and this was a big deal. The B cell people were very happy and very proud of themselves that they had sorted this out, and they had clearly shown the way for other factors. So what you needed to do was to get a bioassay and make it a quantitative bioassay so that you could tell the difference between different supernatants and different RNA and translation product samples. So that was important. But in addition, you needed to be able to get from the DNA to the RNA into the protein. And that was a very critical kind of thing. And this really showed the way going forward. Now, it turns out that in addition to subsequently what happened, a lot of things happened, but I'm just going to touch on a few of them. In addition to IgG1, this interleukin-4 would also cause the switch to IgA as well as IgE. And in the annals of immunology and medicine, the switch to IgE became very important because, as you know, IgE, and we also reviewed the Ishizaka's 1967 paper on IgE as being the protein, the immunoglobulin that's responsible for allergic responses, because it binds to the Fc receptors that are specific for IgE on mast cells, and then they cause the immediate hypersensitivity reaction when they're triggered on the cell surface via the IgE. Turns out if you make an IL-4 knockout mouse, they don't make any IgE. That became very important medicinally because antibodies that will block the interleukin-4 effects onto B cells are effective in really dampening down severe and chronic allergic reactions, such as chronic asthma and also atopic dermatitis and so forth. So it's really added a great deal to that. I mean, before that, the only thing people had were glucocorticoid hormones, essentially. Then I just want to mention, because we're going to get into some of these as we go forward in the Journal Club, in November of 1986, 2 more papers were published about T-cell derived factors that worked on B cells. One from Honjo and Severinson, who now collaborated with Kiyoshi Takatsu, who was from Kumamoto University in the southernmost island of Kyushu. And he had been interested in trying to find out what T-cell replacing factor was. You may recall that we reviewed a paper by Annalisa Schimpel and her professor, I'm blocking on it right now, Weber, I think. Anyway, T-cell replacing factor was thought to replace T-cells totally. Kiyoshi Takatsu in Japan wanted to find out what that was, and he'd been working on an assay for that. And so together with Severinson, helping him to get it more quantitative and so forth, and also with Honjo and his SP6 promoter situation, they cloned the gene for what subsequently became called interleukin-5. Now, interleukin-5 in the mouse does cause B cells to make more immunoglobulin, but it doesn't do that in the human. But rather, what it does do is cause the bone marrow derived cells to make a lot of what we call eosinophils, meaning that these are polymorphonuclear leukocytes that are in the blood and are quite prevalent in 2 situations. One, parasitic infections, sort of like worms that infest you and that sort of thing, and also an allergy. And now antibodies that will block the interleukin-5 response are also effective, and they're being every time I turn on my TV at night to find out what the news is, I'm bombarded by these advertisements for for all these anti-interleukins. And then the other thing that was published in November of 1986 was a paper describing another factor that derived from T cells that worked on B cells. And this factor, the gene was cloned by Toshio Hirano, working at the University of Osaka, together with Tademitsu Kushimoto, who'd been working on these B cell factors for quite some time, for at least a decade. Toshio was a very good biochemist. What he did was, he did what the other people couldn't do. He took protein, generated lots and lots and lots of protein from T cells, and purified it to homogeneity, and then did amino acid sequencing of peptide fragments from this protein. So he generated a bunch of digests of the protein, sequenced them, and then on the basis of the protein sequence, he could make cDNA probes to probe libraries. And that's what he did. And he generated what is now called interleukin-6. So those papers are important, and we'll get into them as time goes on. But that didn't do the whole thing. That still didn't solve the problem, because no matter if you had these factors, even though they would cause B cells to proliferate and they'd make immunoglobulin and they'd do all these great things, they couldn't make long-term B cell lines like we had in T cells. And it wasn't until 5 years later, in the early 90s, that it was discovered that there was a reason for that. And that was that on the surface of T helper cells, there was a molecule that would bind to a molecule on the surface of B cells. And that was a key factor, because that solved the problem of what was called cognate recognition, and initially described by Av Mitchison back in 1971. And I think we went through that paper as well. So we'll get into that. But I think the most important, when I first started doing this journal club, I tweeted about it, and I got a tweet back from somebody who said, we started the journal club in 1798 with Edward Jenner and the smallpox vaccine. And this person tweeted out saying, we only really start really paying attention to papers in immunology from 1980 on, Dr. Smith. So what are you boring us with all these things? And the reason is, as pointed out in all these B cell factors, and there is still stuff that is not understood in terms of what T cell help actually does, whether it causes the class switch and how it really causes the B cells to make huge amounts of immunoglobulin and so forth. So there's a lot of questions that people have been working on for 30, 40, 50 years that are still unsolved. And these are the kinds of questions that if you know your history of immunology, you can start to work on, that the rest of the crowd is sort of rushed on through and forgotten about and don't even know that there are still questions. So I'll stop there, and don't forget to like this video, hopefully, and click on the bell and get notified for more, and go on to my website. So thanks for tuning in.